Okay, hello everyone. We are in Raleigh, North Carolina, in the second day of the 2025 PSA Annual Meeting. And we are with the doctor.
I'm Dr. Scott Kenney.
Okay.
I'm an associate professor at the Center for Food Animal Health at Ohio State University.
Yes.
Basically, I'm a emerging infectious diseases of livestock and poultry specialist.
Yes.
I did my undergraduate work in animal biosciences at the Pennsylvania State University. From there, I went to the Penn State College of Medicine, where I did my doctorate in microbiology and immunology.
Yes.
I went to Virginia Tech, where I did a postdoc with XJ Meng on swine infectious diseases, mostly in vaccinology.
Okay.
From there, I was recruited to Ohio State University. And as I mentioned, I've been there for about 9 years, and I'm now an associate professor, and I work on mostly microbiology and immunology of emerging diseases. We have a large animal biosafety level 3 facility, so we can do high containment research on agricultural animals. And so I work on a number of different viruses, including high-path avian influenza, which I talked about this morning.
Yes.
We work on coronaviruses, African swine fever virus, Japanese encephalitis virus.
Yes.
So we have quite a broad range of viral pathogens that we work on in the animal model to understand how they have pathogenicity and cause disease.
Okay, doctor, that's great. Thank you for that introduction. First, I would like to ask you what is highly pathogenic avian influenza?
Yeah, so highly pathogenic avian influenza virus. Of course, it's a virus. It's an envelope virus. It's in the Orthomyxidiridae family.
Okay.
It is a it is a segmented negative stranded RNA virus, which means it basically has little snippets of RNA, which is what makes up its genome. Depending on the influenza virus, it either has 7 or 8 of these genomic segments.
Yes.
And then they encode for proteins, which then make the virus particle, which then, when it's in a host cell, it makes new virus particles. That's basically everything that viruses do is to make more of themselves. They're then released from that cell, and then they infect a new cell. So that's in a nutshell. So when we're talking about high path versus low path, really it comes down to the hemagglutinin protein, which is one of the proteins that are on the outside of the virus.
The surface, right?
Yep, on the surface of the virus particle. And this HA molecule basically binds to sugars on the host cells.
Yes.
And that determines which cells and which species can be infected by the influenza virus.
Okay. Right.
With high path and low path, so there's a second protein. The second protein is called neuraminidase. Neuraminidase is like this protein that cuts other proteins off of the virus. So when the virus is trying to be released from the cell, it needs to cut and be released. And so, I messed that up a little bit. It really is all in the HA, not the neuraminidase section.
Okay.
So it's during virus fusion.
Yes.
And so basically low path does not have this thing called a polybasic site.
Okay.
So a polybasic site is just these little pieces of amino acids that make up the protein.
Yes.
And if it has a specific cut site, it only infects. It's an enzyme called trypsin, which is located in your upper respiratory tract.
Yes. But
and that cut is needed to help the virus once it binds to the surface of the cell. It's what's needed to help fuse the virus with the cell and allows it to enter into the cells.
Okay.
Low path only has trypsin. The high path has a longer called a polybasic site.
Okay.
And it can also use furin, which is another enzyme. Furin's more recognized with the SARS coronavirus 2, but influenza is the same way. With this furin cleavage site, it can then become more systemic, and it makes it more lethal to poultry. So you have high path, which can basically enter a whole bunch of different cells, then low path, which is restricted to the upper respiratory tract.
Okay, thank you for that great explanation. I hear in your presentation that H5 and H7 are the viruses that affect birds.
Mostly, yes.
What about the others? The other viruses? For example, H1.
Yeah, so typically, as you said, that H refers to the hemagglutinin.
Yes.
And so those hemagglutinins, based on their sequence, will bind to different sialic acid residues. So the alpha-2,3, I believe, is an avian-specific sialic acid. And so if that hemagglutinin is specific for the avian version, it will infect poultry.
But in poultry is 5 and 7, right?
Sometimes. So 5 and 7 are the ones that are typically worried about because of the high path avian influenza, because they can also transmit to humans. So H5, H7, and H9 are actually the high path strains that can infect humans, whereas most of those HA strains, depending on if they infect avian strains or avian sialic acids, they can be infectious to poultry. Most of those other H's are low path because they don't have that polybasic site. But if any of those H's bind to the avian sialic acid receptor and have that polybasic site, they can mutate into a high path influenza, potentially.
Okay, great. And how highly pathogenic avian influenza virus jumps into dairy cattle?
That is a great question. The answer is we're not 100% sure how it got into dairy cattle. It is disconcerting that both the B313 and the D1.1 in 2 separate instances have jumped into dairy cattle. So they were unseparate events. So it appears that, at least multiple transmissions from avian into bovine have happened. What we imagine happening is it's an environmental contamination. So this new strain, this 334B strain of avian influenza has better adapted to mammalian growth. And so environmentally, it can be in all kinds of different places. So it can be in the water supplies. It could be in domestic animals like cats and raccoons and skunks and mice.
And rodents,
In rodents. Exactly. So it can be basically all over these farms. And somehow some of that virus managed to make it from the environment into a cow teat, and then it sort of took off.
Okay, great. You talk in your presentation about the MAT, the highly pathogenic avian influenza dairy infection outbreak MAT. Can you explain that, please?
It's the map that I presented was like sort of a best case scenario of the amount of virus that could be made. So these viruses are amazing in their capability to reproduce themselves. And somehow the mammary gland for cows is probably the best replication site that I've ever found for any type of virus. What we found was when we experimentally infect cows via the teat, you get...
Excuse me, only dairy cows or kind of cow?
So I believe it's been shown that all kinds of cows, but especially dairy cows. I don't know what the titer is that comes out of the dry cows, but the ones that are actively milking, as we showed experimentally, massive titers. So 10 to the 11th, which is like 700 or like billions of virus particles per milliliter. And we're not exactly sure what, it probably has something to do with the physiology of the cells. The cells are there to make, milk globules, which are very similar to virus particles. But wherever that case is, you crank out like massive amounts. And then, the math that I did, I took, a 2,000 cow dairy operation, which is pretty common in the United States. It's about an average size. And if you figure about 1/3 of those cows become infected, that's like 660 of those animals. Their milk production massively drops off. So instead of getting 60 liters a day from these cows, you probably get like 1 liter a day or less. So massive drop off. But what's coming out is super concentrated virus. So you get that 10 to the 11th. And if you just simply multiply, that 660,000 milliliters, because you do the conversion from a liter to a milliliter, order of 3 magnitudes. But by the end of the math, so you do 660 mils times, 700 billion infectious particles per mil, you really get like 460 quadrillion potential virus particles being shed by those 660 dairy cows, per day. That's, an astronomical number. And sometimes that waste milk, a lot of times, dairies will pasteurize it, which is the best way to kill that virus. But some of those dairies just don't have that capability, and they just dump, their effluent back into the lagoon, which you're now setting up this massive virus production factory that could potentially spread the virus.
Okay, doctor. And finally, is vaccination an option?
depends. Like from our first day, the lawyer who gave her a talk, and she was saying it depends. As Dr. Swain was talking about, it should probably be analyzed and used on a case-by-case basis. Vaccines have been proven, safe and efficacious for humans. It will work for poultry, but it should be applied in specific cases where you have to knock down that virus shed. As Dr. Swain said, there's no sterilizing immunity, so you probably have that, at least chance for initial infection. But anything you can do to knock down that overall shed of that virus, it would be important for hopefully controlling that spread to other premises.
Okay, doctor, that's it for today. Thank you for your time. Yeah, and have a nice day and enjoy the rest of the meeting.
All right, thank you so much. It was nice to meet you.
Thank you.